Bio20. In this video, I'm going to be talking about the process of cellular respiration. So we're going to specifically be talking about aerobic cellular respiration, which is talking about when there's oxygen present. In class, I'll also be talking about another type of respiration called anaerobic, but we'll get to that in class. So this regular cellular respiration, when oxygen is present, is broken down into 3 steps. The first step is glycolysis, then we have the Krebs cycle, and then the electron transport chain. So we're going to go through all of the steps and kind of what is happening at each stage. So our very first step, glycolysis. Okay, glyco kind of sounds like glucose. So we're talking about glucose, and lysis always means to break. So if I'm actually breaking down that word and looking at what it means, it's talking about the breaking of glucose. So basically just a series of reactions where we're breaking the glucose, which has 6 carbons, 1, 2, 3, 4, 5, 6. We break it into 2, what I can call either pyruvic acid or pyruvate, that have 3 carbons each. And the reason that we're doing this is because glucose as a molecule is too big, and eventually we'll want to go into the mitochondria. The glucose is way too big to fit into the mitochondria, so we can break it down into ATP, but it needs to be broken down into smaller pieces so we can actually fit into the mitochondria so that we can actually start to do that process of breaking it into ATP. When we're doing this process, there also is 2 ATP molecules and some NADH molecules produced. Just so you know, notice that it doesn't have the P, NADH, it's not NADPH anymore, because P is only included in the photosynthesis, and they both start with the same letter, so that's how you remember that. In cellular respiration, it is just NADH instead. So, just so you know, this happens in the cytoplasm of the cell. Okay, remember it happens in animal and plant cells, and it's just happening before we get into the mitochondria. It happens in that liquid that fills the whole cell. So that is what we're going to be looking at for that first step. Okay, once we have pyruvate, we still need to actually break it down a little bit more, and we have to break it into acetyl-CoA, which is only 2 carbons. So now it will finally actually fit into our mitochondria. And in the meantime, that other carbon that we had left over is creating carbon dioxide, which we do not need, so it's going to be released as waste. This also happens in the cytoplasm. Okay, once we're done the intermediate step, we can take that acetyl-CoA and we can actually put it into the mitochondria, and it's going to go through a series of steps with many enzymes and all this fun stuff, and it's going to create a bunch of stuff for us. So there's a couple things in your guys' notes that I want you to cross out. So cross out a large amount of ATP because it only makes 2 of them in this step. It does create carbon dioxide. It doesn't create water, that's later. And it creates a whole bunch of heat in this process, and also some NADH and some FADH2, okay, which is going to be used for energy storage molecules, just like NADH is, just like ATP. It's an energy storage molecule. And that's what's going to power our third step, the electron transport chain, is these energy storage molecules. So that is the whole point of the Krebs cycle, is just making those energy storage molecules so that at the end we have energy to make a whole bunch more ATP. So that third step, the electron transport chain, it's going to be in the inside of the mitochondria, which the membrane is called the cristae. It's kind of the squiggly line of the mitochondria. Basically, oxygen is sitting at the end of the electron transport chain. It's called an electron acceptor, and it's what's pulling the electrons kind of towards it so that the electrons actually will go across the membrane. And when we're using those NADH and the FADH2 molecules, it breaks it into ATP. One NADH molecule can make 3 ATP, and one FADH2 can produce 2 ATP. So we're going to end up using these and creating a lot of ATP at the end of the electron transport chain. So you guys have this picture on the next page. We're going to start to fill it in, but first let's label kind of what you're looking at. So the outside here is looking at the cell membrane. Okay, all of this would be the cytoplasm. All of the space is the cytoplasm. And in this cell, obviously, there'd be a whole bunch of organelles, but I just ignored them to make it a little bit more easy to look at. So the only organelle I drew was the mitochondria. Inside the mitochondria, that space in the middle is called the matrix of the mitochondria. In between the 2 membranes is called the inner membrane space. And then the squiggly line is called the cristae. So that is kind of the parts of our mitochondria for us. And remember, obviously, there'd be other organelles, but we're just going to ignore them for right now because otherwise it would just be really complicated. And it's already complicated enough. We don't need extra stuff to look at. So now that everything is labeled, we're going to start by writing down our chemical equation for cellular respiration, which is glucose plus oxygen is equal to carbon dioxide, water, and ATP. So we're going to get a glucose molecule, we're going to break it down into pyruvate. We have oxygen at the very end of the electron transport chain to kind of be that electron acceptor and pull it towards it. And then we're creating carbon dioxide a couple different places and releasing it as waste. Okay, we're also creating water at the very end. And of course, ATP is the whole point of this, so that we have energy for any other process in our cell. Okay, maybe we're doing active transport and we need to use energy to do that. Okay, that's the whole point of this process, so that they actually have energy to do whatever needs to get done. So we're going to go through the steps and write what's happening where. So we're going to start with glycolysis. We have our 6-carbon molecule glucose, and we have to break it down into pyruvate. Okay, in this process, we're creating a little bit of ATP and some NADH, which we're going to be using later to power our electron transport chain. Okay, remember pyruvate is 3 carbons, but still not small enough to fit into our mitochondria. So still in the cytoplasm, we have to go into that intermediate step. We have to break our pyruvate into acetyl-CoA and some carbon dioxide. The carbon dioxide is going to leave as waste. Now we have our acetyl-CoA small enough to fit into our mitochondria. It's going to go into the Krebs cycle. Okay, the Krebs cycle is just, you just need to know the products of it. So just what's happening is things being added together, things being pulled apart, but we just need to know the products. So we have carbon dioxide being created. Again, it's going to leave as waste. We do have a little bit of ATP. This isn't in your picture, to like, there's not a spot to draw, so you're just going to have to add it in. So 2 ATP being created. And we also have that kind of sidekick FADH2 being created. We have 2 of those. And we have 6 NADH molecules. So again, NADH and FADH2 are energy storage molecules that are basically there to help us power that last step, the electron transport chain. So remember, the electron transport chain, I'm going to zoom in on it, but it's on this membrane that I'm zoomed in on, the cristae. So like squiggly membrane is where the electron transport chain is. So this part would be the matrix of the mitochondria. This spot underneath the membrane would be the inner membrane space. So we're basically going to be building up that concentration gradient of hydrogen ions in the inner membrane space, okay, to power our ATP synthase, which would be right here. So just again, we're going to take those energy storage molecules, we're going to be using them to power the electron transport chain. Before the electron transport chain, when we talked about it in photosynthesis, it uses sunlight to absorb it. This time we don't have photosystems to absorb sunlight. And we're also sometimes talking about in an animal cell, which we wouldn't be getting sunlight in and absorbing it. So we have to use different energy to power this process. Even though it's a similar process because it's still an electron transport chain, we need energy from something else in order to actually make it go. So we're using this energy storage molecules instead of sunlight to power our electron transport chain in our mitochondria. So I'm going to just zoom in so we can actually see what I'm doing. Oh, but I forgot to mention that heat in the Krebs cycle is also created. Because the Krebs cycle is a super inefficient process, it releases a lot of heat because it's changing one form of energy into another. So that's why heat is always created, because no energy conversion is ever 100% efficient. It always creates heat. But that's why it always, they always say like you have to move around when you're cold, because it's creating heat doing this process, making ATP, so we can move our muscles. So when we're moving our muscles, we have to go through cellular respiration first, and cellular respiration creates a bunch of heat for us. So that's how we warm up if you're cold. That's why you start shivering if you're really, really cold. So now we're going to zoom in on the electron transport chain. At the electron transport chain, we're going to use energy from NADH to energize our electron, make it ready to go, shoot across. Okay, when it shoots across, it's going to allow some hydrogen ions in. Remember, we're building up that concentration gradient of hydrogen ions in the inside of the mitochondria, in the inner membrane space more specifically. Okay, we're going to take some energy from the FADH2 to energize our electron. Now it's again ready to go. Okay, it shoots across. Remember, we built up that concentration gradient, so now it's trying to find any way it can get out of this cell, and it finds a small little narrow passageway through the ATP synthase, squeezes itself through, causes the ATP synthase to spin and create ATP for us. In the end of this process, because we have a certain amount of NADHs and FADH2s, we actually create 34 ATP, which is pretty spectacular. So at the end, That's the whole point, is creating that ATP, but we also create some waste products. So remember I talked about oxygen kind of sitting at the end. It's sitting there being the electron acceptor. It's pulling the electron towards it. So it also will take the hydrogen ion and it will create a bunch of water, which we don't need, okay? We breathe out carbon dioxide, which is the first waste product. And if you guys didn't know this, we also breathe out a little bit of water vapor. That's why when it's really, really cold outside and you go outside, it looks like you're almost smoking. Or if you go to a glass and go against the glass, you get that film and you can like draw a smiley face in it. That's water vapor condensing on the glass. So just, that water is going to be released in our breath and we don't actually use it really at all. So that is the whole process of cellular respiration. Okay, you do need to know the picture that I just drew, the parts of the picture for your drawing quiz. Parts of it are kind of similar to photosynthesis and other parts are not. So practice it a couple times. Hopefully you're ready for your drawing quiz, and if you have any questions, of course, come and see me. That's totally fine. But if not, good luck, and I'll see you guys tomorrow. Good night.